I think Patrick and Ahmed have really set the stage for what Johanna and I are going to talk about next, because really talking about the overarching view of food safety and in terms of policy, in terms of what are the intervention points in the value chain. So we want to zoom in, because one of the things as nutritionists and people who are working in agriculture and nutrition is our key interest is the outcome measure. And as all of us are very familiar, one of the key outcome measures that we are trying to address when it comes to nutrition is stunting in children, infants and young children. As you know, there are about 34 high-burden countries with very high rates of stunting. And many of these countries are also in areas of the world where you find very high burden of food safety issues. So what we wanted to do was moving on from what Ahmed and Patrick were talking about, which is a sort of overarching view, just sort of look at it from a perspective of the outcome measure of interest. And how do you link it to food safety issues? So we're doing two presentations. I'm going to talk about environmental enteropathy and what that means. So we're looking at the individual condition and linking it to stunting, and talk a little bit about what the markers are and why do we see this as an important issue. And then Johanna is going to follow up and talk more about the mycotoxins, and this is her dissertation research, so there's no other better person who can speak about this other than Johanna. Just to move on, I think Ahmed made a comment about, what our global nutrition targets are, and this is a slide from, and I'm sorry, I apologize, this is not the best projector we have here, but these are the global nutrition targets for 2025, and this is a combination of both targets for addressing undernutrition, as in stunting, wasting, and anemia, and underweight, but also the prevalence of non-communicable diseases and hypertension and adult overweight, etc. So these are World Health Assembly. The top ones are the World Health Assembly global nutrition targets. And the key thing is, in these high-burden countries, in Nepal, India, Cambodia, Bangladesh, many of the South Asian countries, a lot of East African, Southern African countries come in that target range, have committed to reduce stunting in their countries by 40%. So that's a very big target that has been put up. And so the key question is, and many of you might be familiar with the Lancet 2013 and those sort of 10 efficacious nutrition interventions to be implemented at 90% coverage. And I can talk at length about what I think about that 90% coverage, but a key set of points that I want to make on that is the focus is on diet diversity, on nutrition education, on knowledge. A lot of it is focused on the diet. There is some focus and some emphasis on nutrition-sensitive approaches, which include wash, hygiene, and sanitation. I think post that particular 2013 Lancet series, there's been a lot of sort of discussion happening around all those nutrition-sensitive approaches that bring agriculture and health to the table with nutrition. And this particular slide, and again, apologies, you're not going to be able to read this, and I'm hoping we can share these with you, but this is called, it's called the stunting syndrome pathway, which was developed by Andrew Prendergast and Jean Humphrey. And I really like to show this because what really emphasizes two things. One thing is, it's not a unidirectional process. We keep looking at the causes of malnutrition, and we go straight, our arrows go straight downwards, but in fact, it's actually a cyclical process. What happens in infancy and early childhood does affect in school age, puberty, and eventually the adult, and then it goes back. So again, this is not something new that I'm presenting here. All of you are very familiar with this. What I want to highlight is, and let me see if I can, because I know we don't have a marker here, and I don't think I can get my mouse there, because for some reason we don't have the same view. But the boxes are basically identifying, on those gray boxes are basically identifying all the different areas or issues of intervention. And the key thing that comes up across all those boxes is EED, which is environmental enteric dysfunction, aflatoxin contamination, arsenic, everything that has got to do with, it's got nothing to do with diet and nutrition. And what I'm thinking is we've got evidence emerging on this, but we don't have enough to sort of say, how do we insert these into our nutrition interventions? So moving directly into what this means, what are these? Let's just look at the concepts a little bit. What is environmental enteropathy, or it's also called EED, which is environmental enteric dysfunction. Now, this was something that was recognized in the early 70s at ICDDRB, which has done some fabulous work on diarrheal disease research. it used to be called tropical sprue, and I'm not the expert here. We've got people at Friedman School and Hopkins who can explain to you why that nomenclature has changed. But what it is it's a systemic and long-term inflammation of your gut. And Patrick called it the leaky gut. That's also a very common way of representing it. And what it is basically your gut is in a perpetual state of inflammation and immune stimulation. And that causes changes in the way your in the gut appears. And I'm going to show that in a second. But over time, that leads to a disruption to your intestinal function. And parts of that, which Patrick has also mentioned, is the issue of nutrient malabsorption. That's a very big issue. But you also have an issue where you have this bacterial translocation happening, and often not really good bacteria that are going into your intestinal lumen. So that sort of takes me to the next sort of set of definitions. We hear a lot about the microbiome, and I thought that really important for us to sort of say, what is the microbiome? So we all carry with us our symbiotic partners in our intestine. We all know that. We all have a certain amount of microflora, and that's the ecosystem of microbes, which we call the intestinal microbiota. And we have a certain amount, millions of them probably, of different species of microbes, and they're really important for us for nutrient harvesting from the diet, to prevent inflammation, to improve the immune response. So there is a certain composition of the microbiome that does exist in a healthy body, and that's really, really important. The question then is, what happens when you are constantly exposed to poor food safety, where you're introduced to enteric pathogens, where you're introduced to heavy metals or pesticides? How does that affect the microbiota? So bringing these two things together, I just want to show you basically the top. This is the intestinal wall, and what you're looking at are called as the villi, and they're kind of like fingers, and they stick up like this, and we all have this. Is the small intestine. And what you see on the top is what it is normally. And when you have altered microbiota, or when you have this enteric dysfunction, it's long-term enteric dysfunction, what you see is what is called as the blunting of the villi. So if you think about your fingers, this is how your villi should be looking. They're like, like they're trubs and crests, and then basically what happens is that they just basically collapse. And here's where the problem begins, this is where nutrient malabsorption becomes an issue. This is also where you're going to have the leaky gut syndrome, where you can have contents of your intestine, which could be really bad bacteria that could get into your body. Okay? So that's just to explain to you in a sort of a graphical way what that looks like. So how do you measure EED? One of the most common tests that I use is called the lactulose mannitol test, and it's considered as the gold standard. But by no means is it the gold standard, because it has a lot of issues when it comes to its specificity and its sensitivity. But at this moment, and we are talking 30 years of researchers working on issues of gut inflammation, that's really the best we have. We've got a whole slew of research teams, whether it's at Johns Hopkins, at Harvard School of Public Health, that are looking at other measures, and I'm going to go through them in a second. But what do we know about the LM test and stunting? What we do have some data from Uganda. This is our own studies. One of our graduate students is working in Uganda, and she has done the LM tests in about 385 children who were about 12 to 16 months. And what's very interesting, these are rural Ugandan children from the southwest of Uganda. So they're really exposed to a lot of different elements in their environment, which includes things like chickens and goats and livestock in their backyard. And what she found was that they had 58% of them had moderate EED, that the LM score that they had was put them into the category of having moderate EED, and 22%, so these are 12 to 16-month-olds, these are kids who are within the 2-year window of your opportunity to prevent stunting. 22% of them had already severe EED. So these are slightly older. It would have been interesting to look at what happens to children as soon as they get off exclusive breastfeeding, but we managed to get them at a slightly older age. Of course, clean water mattered. The households that had clean water had kids with much lower LM scores. And when they had goats and sheep inside the home, and this is one of the things that has been popping up, the whole exposure to feces of chickens and goats and poultry, they got significantly worse. And this is something to think about when you're thinking about a livestock intervention to improve access to animal source foods. There is an unintended consequence right there. And essentially what she also found was the LM score was significantly worse in infants who were wasted or stunted. So we cannot explain causality here because this was just a one-time point measurement, but what it's telling you is that these kids who were already stunted or wasted had a presence of EED at a much higher level than the kids who were not stunted or wasted. In terms of other markers, as I mentioned when I started off this slide, that the LM test is not the most accurate method. It requires, and it's also very intensive because you have to provide a solution to the infant. Just think about if you're trying to get a 12-month-old infant a solution of like 100 ml, which is not a lot, but they have to consume it and you have to monitor them for 6 hours and collect their urine for 6 hours. And that sort of comes to the whole point that often the kind of measurements that we need to do to understand these relationships are not easy to do in the field. The second thing is the urine has to get tested for the ratio of lactulose and mannitol, and we can do that only in the US. So that's another big issue because we've got to ship everything. So then what are the possible other markers? And so one of the things that has been explored are several different groups, including folks at ICDDRB, are exploring fecal markers and other inflammatory markers, which require taking a fecal sample. Our colleagues, Chris Duggan, who's a pediatric and gastroenterologist at Harvard, is looking at blood markers. At this moment, unfortunately, we don't have a substitute for the LM test that is going to be a much more accurate measure and a much more field-friendly measure. So we're really, really at the start of, in the infancy, as Patrick said, when it comes to a lot of these markers. So moving on to the whole issue of the microbiome and this sort of interaction with livestock. Again, this is one of the things that does come up in a lot of our literature when it comes to agriculture and nutrition. So what's the underpinning behind it? And this particular graph explains this. Left side is the gut of a malnourished child. And you're seeing in red are all those microbes that should not be present. Those are pathogens. Most of them are pathogens, and most of them have been found in this particular study, and you have other studies that will describe to you in depth the different types of species you find, and I'm not going to go into that because that's not the purpose of this slide. And on the right side, what you're seeing is the normal child's gut. Now, what's very important here is what you're seeing on the left side is pretty much bacteria that are shared with animals. So this sort of brings it to a circle that, yes, it's important. We have to look at livestock ownership and how livestock products and production in the household can improve the diet diversity through animal source foods, but we need to keep in mind we need to think about good agricultural practices that Ahmed has mentioned and how that we can prevent children who are supposed to benefit from having livestock around from having a shared microbiome. And why is this important? And this is a study that came out in 2013, which is the first one that really sort of highlighted the importance of the gut microbiome. And this is the microbiome study in Malawian twins. We basically were discordant for kwashiorkor, which is bilateral edema with acute malnutrition. That's another way we can use the terminology for that. And what you have is basically one of the children was suffering from kwashiorkor and the other one was not. And they basically studied them in the first 3 years of life, and about 50% of them remained well-nourished. And what you're seeing on the right side is essentially when those kids were introduced, the healthy twins, which are on the left here, when they were introduced to RUTF, they were given RUTF. Both sets of twins were given RUTF, or ready-to-use therapeutic foods. And the y-axis is essentially a score which predicts their microbiome. And the higher the score, the better the microbiome. And what you found is that the twins that were normal actually did very well. And the twins that were discordant did well, but then they started banking down again. So essentially, despite the fact that they received RUTF for the same amount of time, the ones that were malnourished, for whom the RUTF was very important, didn't actually benefit as much as the normal children. And I don't have the subsequent slides on this. This is a very interesting paper if you want to look at it very carefully. But what they did was they took the microbiota from these twins and introduced them in sterile mice to see what's going on. And essentially, the microbiota are different in these two kids. The children who are malnourished have a lot more pathogens in their microbiota. And the ones that were not malnourished had normal gut. And that translated into the sterile mice. So that was an important finding which sort of said, you know, you can provide a malnourished child as much RUTF as you want, you will find some recovery, but they're not going to completely recover and there's going to be a relapse. Similar work has been done to look at microbiome and stunting, because that's the first thing I started with, the stunting as an outcome. And what you do see is, and this is a study that's just been published, and there is less diverse gut microbiota in those kids who are stunted, and it increases with stunting severity. And they've found a specific species. This is a study that used children from Malawi, data from children in Malawi and Bangladesh. And they don't necessarily understand, and that was interesting because they were microbiologists, and they didn't quite understand because this acedoaminococcus species is not very predominant. You don't normally find it. So that was a very interesting thing. And so this is just to sort of point out to you, I apologize, this is not going to make any sense, but what this is telling you is what is the biological mechanism of linking environmental contamination, of the likes that Patrick and Ahmed have talked about, to linear growth impairment. And what we see is that there are two different places. You have nutrient malabsorption on the left, and on the right side is you have this inflammation that occurs and it continues. So it's not just that you're going to have malabsorption in the kids, but you're also going to have inflammation. And what that does in nutrition is it increases your nutrient requirements. So if you have an infection, for example, if you have a cold or a cough or a fever, each and every one of you has a higher nutrient requirements for that period of the infection. And then as you get out of the infection, your requirements drop. Now, we're all healthy human beings here, so we don't actually feel that. We don't immediately hit that unless it's a severe infection. But for kids who are in a continuous inflamed state, what that means is not only are they malabsorbing their nutrients, they also have an increased nutrient requirement. And at this moment, when we look at nutrient value of a diet, we don't account for these increased requirements. This is another framework which is trying to link mycotoxins to linear growth impairment. And what's very interesting is that the mechanisms by which this happens is almost the same: nutrient malabsorption and immune activation and inflammation. So what that's telling you is that the issue that you see with increased EED, environmental contamination, that could be of enteric pathogenic nature, or that could be from mycotoxins, the mechanism by which it could lead to linear growth impairment are very similar. So they're sort of interacting with each other. This is important in places where you're seeing both, and guess what? In most places, you're going to see both mycotoxin contamination as well as enteric pathogens in the food system. So in conclusion, I just want to sort of mention the fact that we really need to consider, when we're thinking about implementing Lancet interventions, that yes, it's great if we can have 90% coverage and try to reduce the burden of stunting, but if you look at the Lancet articles very carefully, it reduces the burden of stunting only by 20%. So we are nowhere near achieving the targets that we have set ourselves by these 10 interventions. And we really should think about the impacts of EED and the poor microbiome and mycotoxins, which Johanna is going to talk about, in negating any gains you might make. So it's not just about the fact that you won't achieve your targets, but you might negate some of the gains you're making because of the implementation of the interventions. So thank you so very much.
We keep talking about aflatoxins, which there's a lot of research on right now. It's kind of the hot topic, but there's other mycotoxins that we should be worried about. And it also, by talking about other mycotoxins, it relates to the case study that we're going to talk about and the discussion that we'll have after lunch. And then I'll just wrap it up by reminding us of what some different study designs that exist, or some study designs that are out there that you might want to consider, both in terms of research, but also when you're trying to think of policy and what you do with findings from the research. So a lot of this has been discussed already, but mycotoxins are the bigger group of toxins. So aflatoxins is just one type of mycotoxin. And they've been linked to a lot of different health issues, So for example, cancer, aflatoxin B1 is carcinogenic. It's declared a carcinogenic type 1, so there's a lot of evidence of its link to cancer. But then there's also these other, like fumonisins, for example, have also been associated with cancer. Mycotoxins have also been associated with neural tube defects and renal diseases and immune suppression. And most recently, which is of interest in our field, especially is with growth faltering. There's some evidence showing that it could be related to that, and there's a lot of research going on as to how it may be related to growth faltering. And Ahmed already talked a lot about pre-harvest and post-harvest and how mycotoxins can contaminate basically at any point of the chain, which complicates the situation a lot, but also gives us a lot of opportunity for intervening and figuring out where we can mitigate those issues. And just to give you a few ideas, of the next slide will show you a few more, but the mycotoxins that are most commonly talked about are aflatoxin, fumonisins, DONs, ochratoxins, but there's many, many more that have been studied within the agricultural field and also the health field now. So this is really hard to read. I know our projector is pretty bad, but it shows you different types of foods that can be contaminated with mycotoxins, and what I want to focus on here is that there's a lot of foods, for example, maize, which is talked about a lot, which can be contaminated with so many different mycotoxins. If you can read, maize is the third food from the left on the second row. Maize can have DON, you can find DON in them, you can find fumonisins, they can have ochratoxins in them and aflatoxins. And then just to complicate it more, each of these toxins can actually be produced by multiple fungi, for example. So aflatoxin has different types of aspergillus that can produce it. So it's just a very complicated situation where it's not just one thing, one food has aflatoxin, one has fumonisin, they're all over the place. And so what this does is it really creates a challenging environment when you're studying mycotoxins. So there's so many different strains, so many different foods, but then there's also the fact that there's a lot of co-concurrence of these mycotoxins. So with maize, you usually have the two really big ones are fumonisins and aflatoxins, and a lot of the research with growth faltering right now has looked at aflatoxins, but very recently saying, well, what about fumonisins? Whenever you have aflatoxins, you're very likely going to have fumonisins. So how do you tease out that relationship to growth faltering? And then just to challenge us even more, there's no biological threshold. So for example, aflatoxin, you shouldn't have any aflatoxin in your blood, but at the same time, even if you regulate it really well, like they do in the EU or in the US, it is a natural toxin. It's found in foods, there's certain amounts that are allowed, you will find it. So what is the dangerous level that we can have in our bodies? We don't know that yet. And then Shibani mentioned a little bit of this, but those really complicated pathways. So how are aflatoxins affecting our health? Is it gut health and so increasing that permeability to these really harmful pathogens? Is it impaired liver function, just immune issues? There's so many different things that these toxins could be doing to our bodies. And how to tease that out is hard. And then also, oftentimes in the foods that you've, when you're studying mycotoxins, it's really hard to tease out poor diet quality. So is it an issue of just nutrients? Because people consuming a lot of maize, for example, are not necessarily, if that's a big part of their diet, they're not eating a varied diet. So is it just an issue of they're not getting other nutrients, or is it an issue that they're contaminated with mycotoxins? And also, there's ethical considerations you want to consider. So we know aflatoxins are really harmful. We know fumonisins have been linked to cancer too, even at low concentrations. But since we don't have that established threshold, how do we tell people what's dangerous? How do we design a research study without scaring participants, for example, that if we find aflatoxin in their blood, we don't know the dangerous level, right? So you just have to be really careful when you're designing either research or interventions to not instill fear, but also not negate the fact that these are dangerous to health. And just lastly, this has already been talked about, it's really difficult to measure mycotoxins in food. So when you're sampling foods, you don't have just one little bag of corn. You usually have multiple bags of corn people are going to be consuming from. The mycotoxins can actually be concentrated in just one part of that grain or food. And so when you sample, you have to make sure you're getting a representative sample of that food. And even so, no matter how hard you try, oftentimes you miss hot spots that there are of the mycotoxin contamination. So this chart's also pretty hard to read, but I just wanted you to focus on the last 3 rows, showing you different biomarkers that you can use for mycotoxins. And aflatoxin is a great example because we have multiple biomarkers for them, and there's different advantages to each. But you have a urine biomarker, for example, which is a little easy to collect, easier than blood, just because it's not as invasive. But the urine biomarker, one of the disadvantages is it only shows you short-term exposure, so what you've consumed in the last couple of days. And we all know that our diet today is very different from what it was 2 days ago. We vary our diets a lot. So that may not be the best measure for chronic exposure, unless you're measuring multiple times, and which unfortunately is invasive because if you're collecting it from women or children, or and no one wants to give you blood unnecessarily. So, but the advantage to the blood measure is that it gives you a measure of the exposure in the past 2 or 3 months. So it's not a perfect measure because you ideally would want to know, well, a longer period, especially because mycotoxins are very seasonal, which I'll show you soon. But at least it gives you that longer time frame. And then there's also a breast milk biomarker for aflatoxin that you could look at. I just wanted to highlight, so we have a study here in Nepal where we're presenting these results on Wednesday if you guys are more interested, but just wanted to show you the seasonality that we're seeing in our study. So we have a birth cohort going on in Banke, and we unfortunately don't have data for those 2 months in the middle, but we do have data for all the rest of the months, and as you can see, there really are really high levels in the winter, pre-winter and winter seasons, indicating that a lot of this may be seasonal, and there's certain foods that we might want to look at that have been stored up to this period for long and maybe been exposed to mycotoxins. So, sorry, and those are the 3 months I was referring to. And then, this might be a little hard to understand, but so if you look at the aflatoxin exposure in the women from our study by percentile, so for example, you got the lowest percentile at 10th percentile and the highest at 90th. In our study, we're seeing that those in the 90th percentile, when they consume peanuts, when they report consuming peanuts once for an extra occasion in the past week, we're seeing a much higher effect of the peanuts on the aflatoxin level. So we know peanuts are commonly contaminated with aflatoxin, but it may be that those who have the highest levels are the ones that are most susceptible. Every time they eat peanuts, they just are that much more susceptible because those levels are so high. And then this is from my colleagues in Uganda. They're also doing a study with HIV-positive women. And they, as you can see the bars on the right, they show that there's a significant association or difference in both the infants and the women that are HIV positive are showing much higher aflatoxin levels. And that's, they measured them during mid-gestation, after delivery in the women, and then the 9-month-old babies. And then interestingly, they also found this, that the infants in the HIV positive group had lower height for HZ scores, which is that link to growth faltering that we keep, that a lot of groups are trying to figure out right now. So just to remind you when we're talking about our case study, just to remind you of what different research designs you can consider. There's pretty much 2 groups. You can do descriptive studies, which don't necessarily try to quantify a relationship, you just try to understand what's going on. So for example, if you do a qualitative study and just talk to people and get a basic idea of the context, and then you can also do cross-sectional surveys to look at prevalence of aflatoxin, for example. So just, you want to see how many people in your sample are exposed to aflatoxin. But then if you want to do something a little bit more analytical, you oftentimes think of a randomized control trial. You say, if I want to see if aflatoxins are related to growth faltering, I design a randomized control trial, give someone aflatoxins and someone not. You can't do this, obviously, because it's unethical. Yeah, so that just reminds us of the complexity of mycotoxin research. There's a lot of ethical issues in it. So that oftentimes is not possible to do RCTs. Although our case study is an RCT, that is a good example of how you can do that to some extent. And you can also do analytical studies instead. So for example, the study we have in Banca is a birth cohort. So it's a prospective birth cohort where you can actually look at the direction of a relationship. So it's not necessarily causal, but you can start looking at and controlling for a lot more other things. And then I just want to remind you guys, so when you're looking at the case study and trying to, when we're having our discussion and you're thinking about how you guys relate to this field and what you would prioritize, just really meeting the criteria for rigorous research design or even intervention. So making sure that you have enough people, so your sample size is big enough to answer your research question. It's really important that you have an effect in mind. So what is it that you're trying to look at and how many people we need for that? And you would also want to make sure that your outcome measures are measuring what they match your research questions. So oftentimes we look at so many different things, but you want to make sure that that ultimate outcome measure is saying, yes, my hypothesis was right or not. Or if you're doing an intervention, how are you going to measure that impact? Also exposure and dose assessments. So what happens first? What's the direction of the relationship? And does it increase with certain doses? So if you have increasing aflatoxin levels, will you see larger effects on that growth faltering? And then also just always making sure you control for confounders. So I spoke about teasing out that relationship with poor diet quality, for example. You have to make sure that you control for diet quality, both for mother's diet quality, if you're looking at a birth cohort, or the infants, and also looking at water and hygiene, which we spoke about before. So making sure that if those are all held constant, do we still see these relationships with mycotoxins?
In terms of how people are selected, how the sampling goes, and especially the fact that the aflatoxin contamination will differ greatly between the dry and the wet season, and doesn't seem like people are distinguishing between that, etc. But there were some, I think, larger issues that came up as well about kind of the general applicability of this type of study for the issue that we're looking at, because it seemed that the thing that they're trying to find out, at least in my very rudimentary understanding of it, is kind of known already. Yeah? That aflatoxins are a problem is something that is really not new. And conducting a huge study on this, costing I don't know how much money, seems like it isn't all that important anymore, especially considering that the link between intake through maize and aflatoxin toxicity or bad health effect is only one of the issues that go on in this system. It's really a much more complex thing. People are eating other things as well. People might be unhealthy for other things. They may be for other reasons. Yeah, there are a whole lot of things here that could potentially be looked at. And if you could kind of imagine the epidemiology, if that's the right term, of this place as kind of a big causal diagrams with lots of links and lots of bubbles and arrows basically linking to each other. We're looking here at only one of those connections, two bubbles and one arrow between them. That's what this study focuses on. And there are lots and lots of other things. And if we were to now move on and design the next gigantic randomized control study for the next arrow, we'll be studying this for the next century or so, and have spent $100 million by the time we're able to advise anybody on what to do. It seems like an incredibly incremental way of finding things out, and in this particular case, it seems like, obviously the reason we do research is that we want to generate new knowledge, which means there needs to be some sort of uncertainty about the thing that we're working on. And I really wonder if the uncertainty about this link between contaminated maize and aflatoxicity is an uncertainty that is large enough to warrant this kind of study. Probably rather not, it seems to me. And I guess what an alternative way of looking at this system could be, is to try and get a better feeling for the complexity and the interrelationships between things. What happens to the maize? Where does it get stored? And what are the potential sources of contamination that happen all through basically the processing chain and this whole system? And what are potentially other issues also that may arise? But and the way to study this is probably a totally different one. And the reason why I was singled out to take on this honorary position here of rapporteur is that we've been working on an approach that could potentially help us get ahead in this, and is where you really look at such an issue here, or ideally an intervention decision, some idea that somebody may have, and you assemble a team of stakeholders, experts, people from this village to help you draw up the implications of this particular intervention, or the kind of the interconnections in this issue, in a conceptual diagram that tries to put everything into a model that actually is important, according to what experts say, according to what locals say, and then move on to basically see what you can make of this, and often. Well, the way we do it, at least, most of the things that are in such a model can be quantified in terms of, not in terms of a precise number. That's pretty much never impossible, even after running this study. But you can always come up with plausible ranges for pretty much everything that plays a role here, like for the amount of maize people eat, or the likelihood that somebody's going to adopt a certain adoption, or whatever it may be. And once you've done this, you can run a simulation that gives you kind of the full range of outcomes that could potentially arise from an intervention you may be planning. And sometimes it turns out in our work, often that's actually enough. You capture the current state of knowledge, and you try and decide, based on this state of knowledge, what do we expect? Sometimes, quite often, that is already enough to compare different alternative scenarios, different decision options, and intervention to a baseline where you have no intervention. Often the state of knowledge is sufficient, and in other cases where it's not sufficient, such an analysis can help you highlight where the knowledge gaps are. So what research is really needed. Like in this case here, it seemed like people were sitting in their office somewhere and decided, well, that the knowledge gap to address is this particular one here. People probably outside of the system are deciding that this is what we work on, without having looked at the system and really assessed what the uncertainties really are. And I think we have to become kind of a lot more strategic in the way we approach such complex things, because all the topics here are really complex, and oftentimes, and here certainly, we approach them as if there was only one variable that matters, one thing we have to study, and that's going to answer the question. When really that's not the case. And to a large extent, I think this derives from the methods we're all used to using. It's the RCTs, it's the regressions, it's the ANOVAs, etc., that were really designed for very simple relationships. They're lab methods, basically, for kind of basic physics or chemistry challenges, but not really suitable for the complex systems that we're all working in. So I think the first step in an analysis should be, maybe it was here, it's just not visible, not reported here. The first step should be getting an overall feeling of what the situation is, everything that really should be considered in some way, and then afterwards deciding where to take our research.
Aflatoxins and stunting, we think that there is a relationship, but maybe it hasn't been quantified, and we were discussing in our group that oftentimes to get any political movement on certain topics, it seems like we have to spend a lot of time and money quantifying their contribution to poor health, especially to development-related health, such as stunting, to make headway. So I agree, though, it's common sense that these are toxins. They have many ill effects on health. So we discussed, I would say that we had sort of two groups of comments. One is about the study design itself, and we found some specific things that should be considered as they progress, and maybe should have been considered and either were or were not, but we're not sure. One could be, of course, theoretically in a RCT, you have, through the randomization process, you don't have any confounding, but in practical realms, we know that that's not true, and many RCTs can be biased by baseline imbalances. So there are ways to, if you're going to do a baseline in our RCT, there are ways to control for that, such as covariate constraint randomization, and those can be quite important to measure and make sure that you have these balances at the beginning. we discussed that stunting, to detect a difference of 30% between intervention and control in stunting was very ambitious, and that they may be setting themselves up for problems down the line, because we can't even achieve a 20% reduction implementing all 10 Lancet interventions, so that may be a problem. Also, we were a bit unclear about the duration of the intervention and the effect of other foods in the diet, maybe the complex interactions between those foods or complementary foods that are fed to children as they grow up, and if they're similar across both study arms, that won't be a problem in their actual design, but if there's some kind of differential consumption or loss to follow-up in migration or other things, that could be an issue. There's also, of course, with any RCT where you have a known harm to the community that you're trying to mitigate, there's an ethical dilemma of leaving the control out of that intervention, so not providing any testing to the control arm. And I think also that is another study design issue, because if you really want to quantify the link between stunting and aflatoxin exposure, you do have the biological measurements in the children, but you may or may not be able to fully quantify it if you're not measuring the food, the aflatoxins in the food of the control arm. We also found the seasonality issues, and also there could have been adherence problems, and of course if you're doing an intention to treat analysis, you're going to have a possible washout of the effect based on contamination leakage between the clusters, or people potentially hiding food and not having an equal swap, or similar or trading in between clusters and households, things like that. So then the bigger issues that we talked about in terms of the importance of aflatoxins, one of them is trade. So we talked about how you politicize this problem and get how you bring this type of science up into the implementation and policy realms and get governments to pay attention. And one way that we discussed was through trade, whereby, for instance, Africa trades a lot with the European Union, and the European Union requires very strict standards. But this may benefit the system as a whole by increasing control, or it may actually harm the most vulnerable by trading down, where the best products are exported, the healthiest products are exported, and the rest is left there. So there's also complex issues around local production and purchasing and how that relates. So we discussed also, for instance, ways that communities can sort of be in control of measuring and dealing with their own exposure, possibly the development of low-cost test kits and certain initiatives, community initiatives for storage and dealing with these sorts of issues. And we also certainly acknowledged that in the complex food environment, the very sort of linear quantification of mycotoxins or aflatoxins and stunting also doesn't address many other components of stunting, and whether it be diarrhea, open defecation, breastfeeding practices, complementary foods, hygiene, for instance, the pre-existing health condition of the mother potentially could have quite differential effects on how they deal with aflatoxin exposure.